Hello and welcome to Module 5, where we're going to consider Chapter 7, entitled The Blueprint of Life from DNA to Protein. In this chapter, we'll first consider the characteristics of DNA and RNA. From there, we'll look at DNA replication, considering the process by which we initiate replication and how replication takes place step by step. Thereafter, we'll consider gene expression in bacteria, for which there are two primary steps, transcription and translation. Sandwiched in between this, I'll introduce to you three different types of RNA molecules: messenger RNA, transfer RNA, and ribosomal RNA. We'll consider the genetic code, as it helps us in the process of translation and putting amino acids together into a sequence to create a chain. And we'll see how the ribosome is designed to help us do this with the help of tRNA. We'll review briefly the four levels of protein structure we considered at the very start of the term to gain a better understanding of the events immediately following translation. And from there, we'll look at quorum sensing, an example of controlling gene expression and how bacteria might do that. So how are DNA and RNA different? First of all, we look at structure. When we look at the structure of DNA, we see that it's double-stranded and we create a double helix. In contrast, RNA is typically found single-stranded. In terms of the components making up the nucleotides, in DNA we have a deoxyribose sugar, some bases, and a phosphate group. In contrast, we see a ribose sugar. So we have removed an OH group here, and of course we still have our bases and phosphate groups. So we see that there's a difference in that fructose sugar, that 5-sided ring, in terms of one OH group. We're going to find that that's going to change the stability of our RNA in comparison to DNA. We have differences in bases. There are 4 bases associated with DNA and 4 with RNA, 3 of which are shared: adenine, guanine, cytosine. We see that here, adenine, guanine, cytosine. However, when we look at the basis for DNA, we see thymine, and that's been replaced by uracil and RNA. In terms of function, DNA functions with the ability to replicate genetic material so that we can go ahead and create new cells, as well as we store genetic information here to be used for the process of gene expression. When we consider RNA, in whatever context we're looking at RNA, let's say we're looking at an mRNA, a messenger RNA molecule, we're going to see that that helps convert genetic information that's found in our DNA to a functional gene product, typically a protein. Now I'm going to skip location for a moment. We're going to move on to stability. In terms of DNA, DNA is very stable. We need to keep our genetic information safe. In contrast, when we consider RNA, RNA is more reactive. It has the ability to break down much more quickly. And that makes sense because as we make an RNA transcript or use to create a protein, we don't need that transcript sitting out in the cytoplasm long-term to be continued to be translated again and again and again and again and making more gene product. Our cell only wants a certain amount of gene product, and then we need to get rid of that mRNA molecule. So we see that that breaks down. Lastly, in terms of location, the DNA molecule, so we have this round circular chromosome found in bacteria. Remember, we don't have membrane-bound organelles. So rather than nucleus, which we might see in a eukaryote, we see what's called the nucleoid. It's just an area within the cytoplasm holding the circular chromosome. In contrast, when we look at our RNA, RNA is going to, of course, be created in that nucleoid region, but then it's going to be spread out throughout the cytoplasm so that it comes in contact with the various ribosomes within the cell. There are some general definitions I want to cover before we move forward. I want to talk about genetics. Genetics itself is the science of heredity. How does a trait pass from one generation to the next? We look at genome. Genome includes the entire library of genetic information that's stored in a cell, which includes, for bacteria, chromosome, as well as in many bacteria, it also includes plasmids, which are extra chromosomal bits of DNA, and we'll cover that in more detail. We have the chromosome, and that's, again, in bacteria, typically a single circular structure found in the nucleoid that contains all of the genes necessary to make gene products for the cell. Genes Genes themselves are segments of DNA that typically code for some kind of a gene product, typically a protein. And then we have plasmids. Plasmids are commonly found in the microbial world, and like chromosomes, they're double-stranded molecules. And different than our DNA, our chromosomal DNA, we find that plasmids tend to replicate independent of chromosomes. They don't encode for information that's essential for life, for the cell, but they help provide bacterial cells with the ability to cope with a particular environment, which tends to then be harmful for humans or other hosts. Let's look at DNA replication. DNA is copied such that as cells divide by binary fission, the genetic information held within the chromosome is passed along to identical daughter cells. With this in mind, each strand of double-stranded DNA serves as a template for the synthesis of its complement, and we'll see how that works in a moment. Let's go ahead and see how the process of DNA replication works. Let's imagine we're working with a circular chromosome of E. coli, and it could be any bacteria or nearly any bacteria, but we'll just say E. coli. There are proteins responsible for initiating DNA replication that recognize an origin of replication, this short stretch of DNA that has a very specific sequence of nucleotides. And we're going to see those proteins are going to attach to DNA, separate the strands, opening up what we call a replication bubble. Replication of DNA then proceeds in both directions of this circular chromosome until the entire molecule of DNA is copied. Okay, so let's go back to the process specifically now, looking at each, like I have this list here of DNA helicase, single-stranded binding proteins, primase, DNA polymerase, ligase, and some other things. Let's go ahead and look at what each of these proteins do. So at each end of replication forks, we're going to have origins of replication in multiple places. We have this Y-shaped region right here where the parental strands of DNA become unwound. Different proteins participate in this unwinding process to ensure the coordinated action of a group of enzymes and other proteins. So first of all, we're going to have DNA helicase, and that's represented right here. So DNA helicase arrives to the origin of replication first, and it's going to unwind the double helix. Two strands are now separated and can serve as a template strand. Each one serves as a template strand for our semi-conservative replication process. So here was our first strand, 3' to 5', and we're going to see a second strand of our original strand of DNA on our circular chromosome in the 5' to 3' direction. So we'll be relying on one of the original strands to make a complementary strand. And in this way, we have semi-conservative replication. Half of the new strand is the original strand. We have the replication fork, which is kind of this area right here within. We see that the DNA helicase within, and then this is our replication fork. We're going to see that we move along in this direction here from 3' to 5'. It could be 5' to 3', depending on how you look at it. Ultimately, that replication fork is going to be responsible for attracting some things. First of all, we're going to have single-stranded binding proteins. These are proteins that come in and stabilize each of these strands of DNA to prevent something we call reannealing. So reannealing is just the action by which our bases here and here are going to be attracted to one another, and they naturally want to come back together. So the single-stranded binding proteins are going to keep our strands separate such that we can then have other proteins, enzymes, come in to replicate our DNA. We need a primer to help attract our major machinery, that of DNA polymerase. So we rely on an enzyme called a primase that sets down these short strands of what we call RNA primer, segments of a handful of base pairs, which ultimately allow DNA polymerase to bind such that replication can begin. Without primase setting down a primer, DNA polymerase cannot come in. And so our primers are highlighted here in green. And primase is the enzyme which lays down the primer, which is a short RNA nucleic acid sequence. The next step then is DNA polymerase and its arrival. Ultimately, it interacts with the primer we've just laid down, connecting to the template strands. And as it moves along the template strands, so we're going to see these represented in purple. We'll see this one move in this direction from 3' to 5', and this one here also moves 3' to 5', so we actually start here and move in this direction. So we're going to see them move along the template strands, adding nucleotides complementary to that of the template strands. I want to make a note here. Replication always starts in the 3' end and moves along toward the 5' end. I can't emphasize that enough. We call this the reading of a strand. And in turn, when we're creating a new semi-conservative strand, we're laying down nucleotides in the 5' to 3' direction. So if this is our 3' to 5' direction, where we're sending our DNA polymerase in this direction, it's going to be laying down nucleotides here, and this would be 5' to 3', because remember we have those anti-parallel strands. So we see here 3' to 5' going left to right. We can just keep adding nucleotides. It's really easy with this strand here because we already are in this 3' to 5' direction, and our DNA helicase is moving along this way, opening up further bits along here. That's pretty easy, but notice DNA polymerase down here is going to go in this direction to our left because we have 3' to 5' here. And helicase is opening things up here. And so you can see that there might be a problem, right? We're going to run out of template while this is continuing to open up, and we don't know what to do with that. What needs to happen on this strand, we call this our lagging strand. And what happens with this lagging strand, we need to make short pieces of semi-conservative DNA. And in doing so, as the replication fork opens up further, so we'll see helicase opens up our replication fork further, we're going to see another or a new DNA polymerase come in to create another short piece of semi-conservative DNA. And we call this process discontinuous as compared to what we see up here in what we call our leading strand. The new segments of double-stranded DNA we make on our lagging strand, those discontinuous segments, are called Okazaki fragments. I want to emphasize that, Okazaki fragments. They're found only on this lagging strand. I want to take a moment to label these again. This is our leading strand in the 3' to 5' direction. It works out really easily to create this complementary strand in this semi-conservative nature, but on our lagging strand we have some difficulties and we need some additional things to help us with this. So how are we going to create these continuous pieces from a discontinuous production? We're going to rely on a couple of things called ligase and RNase H, or it's also called DNA polymerase 1. So RNase H, or DNA polymerase 1, this enzyme's responsibility is going to be to remove these primers, and we see it work up here too on occasion, but we only produce one nice long continuous strand here, so there's just a little bit of work here for that RNase H. But we are going to need it pretty regularly here on our lagging strand as we're making our complementary strand and piecing those Okazaki fragments together. We have to remove the primer originally set down with primase as DNA polymerase approaches. The polymerase 3, that DNA polymerase 3, can continue moving forward only until it reaches another part of that strand. So it can continue moving in this direction until it bumps up to something green. And then we're going to rely on RNase H to take this primer out. And we're going to see that we're going to put down a couple of new bases here. And then we're going to unite these two strands, blue and blue, as we see DNA polymerase moving this way, with an enzyme called DNA ligase. DNA polymerase is unable or incapable of joining two Okazaki fragments together on its own. We rely on this enzyme called DNA ligase, which is ATP dependent, so we need some energy, and we're going to see that that's going to join the little pieces of these sugar phosphate backbones of this small, discontinuously produced strand of DNA together, creating a continuous strand. Lastly, there is one other enzyme to talk about. We have DNA gyrase, or gyrase, depending on who you hear it from. This is responsible for temporarily breaking up strands of DNA. So ahead of this, as we are unraveling here, we actually create some supercoiling events in the circular chromosome. And so DNA gyrase is going to come in, temporarily break up strands of DNA to help relieve the supercoiling tension that's caused by unwinding of two strands of DNA in a circular chromosome. So I want you to keep an eye on the prize here, so to speak, kind of wrapping all this up together. We have just completed DNA replication. The purpose of DNA replication is to duplicate double-stranded DNA before a cell undergoes cell division. Undergoes binary fission is what we're looking at when we're talking about bacteria, thereby ensuring genetic information is passed along from one generation to the next, identical genetic information. As we consider bacteria, we're thinking of binary fission. Don't forget that. So we want to ensure there's duplication of genetic material before this enlarged cell undergoes splitting into two identical daughter cells. And you're going to see in our Canvas module, I will add an animated video to this showing you how this process works in real time. And that'll help you visualize all of these activities, all these enzymes as they work at this position on the chromosome at the replication fork. Now, let's talk about gene expression. What is gene expression? Ultimately, gene expression is defined as a two-step process of transcription and translation required to make some kind of a gene product, including proteins. So what we just looked at is duplicating DNA. But now we're going to take DNA and we're going to make a gene product, typically a protein. And you see I've mentioned the central dogma of molecular biology here. What is that? Well, the central dogma simply is a term for the process of taking DNA, transcribing it into a single strand of messenger RNA, taking that transcript and translating it into a series of amino acids, ultimately going on to make a protein. So we're going to see this again, DNA to mRNA, messenger RNA, mRNA to an amino acid chain, which through some post-translational activities, we'll see protein made. Now, let's go ahead and define these. Transcription. Transcription is the process that occurs in the nucleoid. We're talking about prokaryotes. So we don't have a nucleus, we have the nucleoid, by which we take the DNA template and synthesize a single-stranded mRNA transcript, which ultimately finds its way to the cytoplasm, by which we're going to encounter free-floating ribosomes, and there we undergo the next step called translation. Translation is the process by which we take single-stranded mRNA transcript that we made in the nucleoid, and with the help of ribosomes found in the cytoplasm, we create a very specific sequence of amino acids that can go on to be modified into a protein. In short, translation is where we use an mRNA transcript to make protein. Let's begin transcription by looking at this process. And this is just an overview. So we have a three-step process of transcription, which includes initiation, elongation, and termination. Remember, in eukaryotes, this occurs in the nucleus, but in prokaryotes, it's going to occur in the nucleoid of the cytoplasm. And we don't have membrane-bound organelles, including we don't have a membrane-bound nucleus. So 3 steps: initiation, elongation, and termination. So here we have this segment of DNA. It's a very informal segment of DNA, but a segment nonetheless. And it holds a gene. So we're going to say this region here, this coding region, there's going to be a gene that contains information needed to transcribe an mRNA transcript, which will ultimately go on to code for a protein. And we're going to see some terms upstream and downstream. Can you guess what those might mean? If this is the region of a gene here, and we want to encode something here, this is our coding region. If we want to just transcribe this, we want to move in this direction. We call this the downstream region here. Kind of like water moving downstream. We might come across a log in a stream, and anything that goes beyond that, moving with the water, is downstream, but anything above that is upstream. Maybe it's a dock in the river. Anyway, so upstream and downstream, you should recognize that. This is our coding region, and then upstream before the coding region, we have other things, and we'll talk about those other things. All right, at the moment, keep in mind, we're looking at a part of a double helix. Now, this is a very linear form with which I've written this out, but this is part of the double helix. And I want you to focus now on this section right here called the promoter region. We call that the promoter, but it's a promoter region because this is where the initiation process begins. And within the promoter region, we have a promoter. And that promoter is typically a very specific sequence of nucleotides. In this case, we're going to see a sequence of bases being held by those nucleotides. We call those TA-TA repeats. So thymine, adenine, thymine, adenine. Sometimes we call that the TATA box. And that's common in many promoter regions across the animal species. So what's the significance of this sequence within the promoter region? That sequence within this promoter region, potentially TATA, is recognized by transcription factors. And I'm not going to get into the specifics of the various transcription factors. But ultimately, transcription factors are proteins that recognize and then bind to the promoter region of a DNA strand in order to initiate transcription. Transcription factors help ensure transcription is selective and efficient, that we're only expressing a gene or group of genes that need to be expressed in a bacterium at a given moment. Transcription factors know where to go, what to bind to, because they respond to various internal and external environmental cues, determining what proteins might need to be made at a given moment. And so this is the very first step of initiation. Transcription factors recognize the promoter region within the promoter, they gather together, and they create what's called an initiation complex. And it's this initiation complex that ultimately attracts an enzyme called RNA polymerase. which is involved in the elongation phase of transcription. And we're going to see then 2 strands of DNA unzip, and we see that unzipping right here, creating what we call a coding strand and a template strand. But I'm getting a little ahead of myself. That belongs to the elongation phase. So back to this initiation phase. There's another area here that's called the activator repressor area. And I want to briefly just mention this. This region close to the promoter region, this region is a site for the promotion of the binding of transcription factors and RNA polymerase if transcription of a given region of DNA is needed or desired. Or it could be for the discouragement of the binding of transcription factors and RNA polymerase when transcription of a region of DNA isn't necessary or isn't needed. Various proteins bind to this region in one manner or another to either promote or to discourage gene expression. And it's necessary. This is necessary because protein coding genes are expressed at different times in a bacterial cell's life, different phases in response to different signals. And in doing that, we're going to say that there's even one more step upstream called the enhancer region, where certain proteins bind here. And this, so we have activator repressor region, but we have an enhancer region. So when we want to activate, we actually might see the enhancer region also on high alert where certain proteins are bound here. And this will further encourage transcription factors and the RNA polymerase enzyme to bind so as to form the initiation complex. So what do we need to know about initiation? First, there's a section of DNA found upstream called a promoter region. And in that promoter region, there's a special sequence by which transcription factors come in and bind. And once transcription factors arrive and bind to the promoter region, this will draw RNA polymerase to the area, setting off the various steps needed for elongation. Now, I haven't mentioned the sigma factor. The sigma factor is actually a portion of the RNA polymerase molecule. And so we have RNA polymerase, but within RNA polymerase, there's a little piece that's actually going to be responsible for recognizing the promoter. And so it's the sigma factor of the RNA polymerase that recognizes the promoter. And then this transcription bubble, once we have all of these components here, we're going to see we create this transcription bubble and unwinding of the DNA will occur. Looking at the next step of transcription called elongation. So this is where we're actually making our mRNA transcript. And we see a few things have taken place. First, we formed our transcription bubble. And in doing so, we've gently pulled apart our 2 strands of DNA in our bacterial chromosome so as to expose DNA to the machinery, those enzymes that are needed to transcribe the mRNA transcript. And it's important to note here, now we're going to talk about the 2 strands. Each of our 2 strands of bacterial DNA have their own unique names. We have a strand that runs in the 3' to 5' direction. And that's what I've drawn here, but here we see both of our strands. So 3' to 5' direction. That's called our template strand. Sometimes it's called the antisense strand or the minus strand, but it's called the template strand, which RNA polymerase binds to so as to form the mRNA transcript. But we also have this 5' to 3' strand, and that's called the coding strand. The coding strand is also referred to as a sense strand or a plus strand, and the function of the coding strand is to protect the exposed region of DNA while RNA polymerase is moving along and transcribing it. And it also serves as a helpful tool for microbiology students when doing homework or study guide materials, because even though the coding strand isn't directly involved in transcription, we're ultimately making a complementary strand to our template strand as we form an mRNA transcript. And this is also a complementary strand to our template strand of DNA. Thus, the coding strand will resemble very similarly the overall mRNA transcript sequence we're making in the elongation phase, except because we're making RNA instead of DNA, its bases will involve adenine, uracil, guanine, and cytosine. We replace thymine with uracil here. Other than that, those strands are going to be very, very similar, and it's an easy way to check to make sure you've done something correct. And so where does the enzyme RNA polymerase actually come in? Remember, we read up and write down. So we read from 3' to 5'. We're reading up and we're writing down from 5' to 3'. So we want to be looking at and looking for this 3' to 5' strand. Don't confuse that with our coding strand. We want to be working with our template strand with our RNA polymerase. And I do want to point out, sometimes the names of enzymes tend to be a bit confusing. With DNA replication, we had DNA polymerase. Now we have RNA polymerase. The best way to keep these straight: DNA polymerase is involved with DNA replication. RNA polymerase is involved with making an RNA transcript. Don't let the polymerase term confuse you. Now, the third and final step of transcription is called the termination step, and that's exactly what happens. This step happens only after RNA polymerase moves down the template strand of DNA in the 3' to 5' direction and makes the mRNA transcript. So, when does RNA polymerase know when to terminate transcription? Generally, RNA polymerase will approach some kind of a sequence-specific termination, indicating to RNA polymerase that it's arrived to the end of a region and transcription should stop. Sometimes it's a terminator sequence, and it's heavy in GCs, guanine and cytosine nucleotides. Alternatively, RNA polymerase might run into a termination protein. There's one that's called Rho. We call this Rho-dependent termination. And binding to this specific sequence of RNA near the end of the mRNA transcript, Rho pushes RNA polymerase off of our strand and lends to the breakdown of the transcription bubble itself. So there are a few ways that transcription may occur, and certainly it's more complicated than this. I'm certainly making it more generalized. Up to this point in transcription, we've seen messenger RNA. We've just made an mRNA transcript, which the transcript is responsible for carrying the code needed to synthesize a given protein, with help of ribosomes and the process of translation that we're going to look at in a moment. As we move along through the remainder of the chapter, we're going to encounter two other RNA molecules. We have transfer RNA molecules, or tRNA. Now, to understand tRNA, I need to explain something to you just a bit deeper about our mRNA transcripts. The genetic information transcribed in an mRNA molecule can be thought of as a series of 3-letter words. They're sequences of 3 nucleotides along the mRNA chain specifying for a particular amino acid. These 3-letter words, so to speak, are called codons. Okay, how does that come into play? So, how do tRNAs come into play with this? This is a tRNA right here, transfer RNA, and we have 2 ends to it. At one end of every tRNA molecule here is a site for which an amino acid attaches. And at this opposite side here is a group of nucleotide, or I guess bases, called the anticodon, which constitutes the point of contact with the mRNA transcript. So the anticodon is going to go look for the codon. And each tRNA species has its own distinctive anticodon, allowing it to unite by complementary base pairing with one and only one codon. And somewhere at some point in time in my own studies, I found a nice little descriptor for tRNA. Ultimately, the tRNAs carry amino acids. They bring them along to the ribosome. They associate with mRNA transcripts, and of course, they're interacting with the ribosomes. So keep those 3 functions of our tRNAs in mind. Lastly, we have ribosomal RNAs. Ribosomal RNA is responsible for making the various parts that comprise a ribosome, which is where translation takes place. Now we look at the genetic code. To understand the next step in gene expression, to understand translation, keep in mind that we're going to be assembling individual amino acids together into a string or a chain, a process we call polymerization, from information we take from the mRNA transcript that's read by the ribosome and helped along by our tRNAs. And so you might ask yourself, what and how do we know what this nucleotide sequence in DNA is going to code for in terms of our amino acids? Well, if we look at this image right here first, we see our template strand of DNA in blue. So we see that in blue. That's where we started quite a while back now. Remember, from transcription, RNA polymerase moves along the template strand of DNA to create a single strand of mRNA that's complementary to the template strand of DNA. So we see that. And with some we're going to see a bit of couple little steps by which we leave the nucleoid and now move out into the cytoplasm formal. We're going to see then some free-floating ribosomes that are going to associate with this messenger RNA. And ultimately, we're then going to see the mRNA transcript. We're going to read it as a ribosome. For a ribosome, we're reading it, and we create a chain of amino acids from the information which it reads. So how does the reading process work? Keep in mind, we have the ribosome. So here's a ribosome right here. And here's our large subunit. Here's our small subunit. We're going to see that the mRNA transcript is going to read in sets of 3 nucleotides. We call them triplets. We call them a codon. Triplets and codon can be used interchangeably, but the more formal word is codon. A codon is defined as a 3-nucleotide sequence within the mRNA transcript that codes for a specific amino acid. If we look at our mRNA molecule here in orange, if we're looking at this right here in orange, we see a 3-sequence AUG, a UUU, and a CGA. These are our triplet codons. We have 3 triplets here. Each codon is a 3-unit code that originates from the DNA molecule where it was first transcribed. Now, to illustrate how this all comes together, let's go ahead and we're going to look at our tRNA molecule. So here's a transfer RNA, and here's our anticodon, here's our amino acid. Remember that we are looking for an anticodon that matches up complementary to our codon. In this case, we want to find something that's complementary to AUG, and A binds as a complement with T, U is going to match up with A, and G is going to match up with C, complementary. So we're looking for something, an anticodon with UAC, and in fact, that's what we have here. So UAC, the anticodon, matches up with the codon, and we know this pairs, and so this is ultimately going to code for something we call methionine, and specifically, we're going to call it FMET. And we can take these same steps for our next codon, which is UUU. See, here's our mRNA transcript, second codon, second group of 3 nucleotides, UUU. What do we pair U with? Well, U pairs complementary with A. So our anticodon associated with a tRNA molecule should be AAA. And we're going to find out that AAA is going to code for phenylalanine. And then we can go ahead and look at our last codon here, CGA, where C do a complementary pairing with G. G would pair with C, and A would go ahead and pair with U. So our anticodon on our tRNA in this third codon position is going to be GCU, and that's going to pair with arginine. So let's look and see how these work. So if we look up AUG as a codon, here are the codons that are associated with our genetic code. If we see A, we're going to be looking at this column here for A. We would be looking, the second letter is up here on this x-axis here, so we'd be looking for U. And then we find our third letter is associated with G. So we have AU and then G here, and that's going to code for methionine, just like we said. Next, we look up UUU, which is pretty easy. It's actually right here. U, and then if we slide over from the third position to U, that's going to end up coding for phenylalanine, which we mentioned. And then lastly, CGA. If we go to the C spot, and we move over to the G spot and we look at the A spot, that's CGA, arginine. So those are 3 amino acids associated with these 3 codons. I'd like you to practice a couple on your own, looking at codons AAG, CAU, and GUC. And the next slide will go ahead and show you the answers to those, but try to work those out on your own. You will have exam questions like that. Now, let's look at amino acids. Every time a new amino acid is delivered, our ribosomes and enzymes associated with the ribosome create a peptide bond between amino acids through a process we call polymerization. We're forming a polymer of amino acids, or you might call it a polypeptide chain. In this process, and with the help of some ATP, we create some covalent bonds between the amino groups of one amino acid and the carboxyl group of another amino acid. And so we see that here. We are going to see one amino acid here, and here we have a carboxylic acid group that's going to bind with the amino group of another amino acid. Generally, an amino acid is a building block of proteins, and there are 20 amino acids found in our body. Each amino acid has the same fundamental structure, which consists of a central carbon right here. We're going to have that bound with an amino group, this NH2 group, a carboxyl group here, COOH, which is an acidic group, and then a hydrogen. Our last structure we call the R group. This is what defines what an amino acid is going to be. This structure here is the same for every amino acid, but we have a different R group depending on which of our 20 amino acids we're considering. The name amino acid is derived from this amino group and the carboxyl group, so amino acid. This is the acid part. Every amino acid has then this R group that's a little different. Sometimes it's referred to as a side chain, and we're going to see that that side chain or that R group, whatever those molecules are attached to that central carbon in that position, is going to give our amino acid and then the subsequent protein some kind of characteristic. Might include size, polarity, or pH. Let's go ahead and look at the ribosomes and their structure. Ribosomes are composed of two subunits. In prokaryotes, we have a large 50S subunit and a smaller 30S subunit, and they only bind together during protein synthesis, creating this sandwich-like structure surrounding mRNA. The purpose of the ribosome is to read the codons of the mRNA transcript and recruit tRNA molecules with the right anticodon to generate a strand of growing amino acids. To do so, each ribosomal subunit has 3 binding sites for tRNA, designated an A site, which we see here, a P site, which we see here, and an E site here. And I like to think of this as an arrival site. This is where the tRNA arrives first, and then it moves to this middle site, the P site. We call that the processing or the pass-through site, and then here to the exit site before the tRNA moves on its way to be recycled. So there are 3 different sites on our ribosome structure that we'll consider. Let's go ahead now and dig into translation. I've alluded to translation being the step of converting our triplet codons of nucleotide sequences found in the mRNA transcript into corresponding amino acid sequence. And ultimately that's going to produce a protein. I've given you a lot of information, so this next step in our lecture should be pretty straightforward. Similar to transcription, translation is also a 3-step process by which we have initiation, elongation, and termination. Remember, this is happening in the cytoplasm. We've been in the nucleoid region, and now we're just free-floating in the cytoplasm such that our mRNA transcript can come and encounter the ribosomes that are floating in the cytoplasm. Remember that as we move forward, recall the ribosome has 3 tRNA binding sites. So again, the A, P, and E sites, arrival, processing, and exit. And most important at the moment is going to be the A and P sites. Up through this point, we had a tRNA in the A site as we recognized the start codon with the tRNA bringing in methionine. So we saw that methionine will now move from this A site where it would have encountered its codon and delivered its amino acid. Now it's going to move on to this P site, and it'll bring the amino acid with it in this particular circumstance. Methionine is now going to be in this P site. We are going to see the next anticodon match up with the codon. We see UGG, so there will be a tRNA molecule with the anticodon ACC arrive to the A site holding tryptophan, and methionine will move from P site to the A site. So the actual amino acid methionine is going to move from P site to the A site. So we're not talking about the tRNA, but just the amino acid. That's going to start the growth of the peptide chain. The original tRNA that was holding methionine will now enter the E site, so the tRNA moves to the E site, and then it's going to exit the ribosome. The tRNA holding tryptophan and now methionine too will move from the A site to the P site, allowing the ribosome to translocate again and read the next codon, which is UUC. It's holding an amino acid for phenylalanine. We're going to see with this next tRNA, UUC will pair up with AAG, and that holds phenylalanine. And that's going to continue the growth of our peptide chain. These are the steps of elongation. Ultimately, as we approach the end of the mRNA transcript, one of 3 stop codons is going to enter the A site of a ribosome. There's no tRNA molecule that recognizes or binds to these codons, so we don't have an anticodon. So the peptide and the tRNA in the P site break apart, whereby the polypeptide is released into the cytoplasm and the small and large ribosomal subunits dissociate and fall away, looking for their next mRNA transcript. I want you to know the stop codons. There are 3 of them. We had 1 start codon, but now we have 3 stop codons. We have UAA and UGA as well as UAG. And there's a nice little mnemonic device to help you remember these 3 codons when you come upon them. UAA can be remembered as you are annoying, UGA, you go away, and UAG, you are gone. I didn't come up with those. Those are things I've known for a number of years. Similar to what we saw with transcription, there are going to be just a handful of activities associated with the maturation of our polypeptide chain into a protein. At the end of translation, we have a primary protein structure made up of a sequence of amino acids. And we're going to see by processing, we'll see some interaction among amino acids, creating a secondary structure that is the folding into either an alpha helix or a beta sheet, and then a tertiary structure, whereby the R groups of the amino acids within this chain begin to interact and create a three-dimensional structure. If we're looking at more than one polypeptide chain bound together to create a protein, then we have a quaternary structure. The quaternary structure is the folding and interactions between two or more polypeptide chains, making up a protein. Our final conversation is involved with quorum sensing. This is a method by which bacteria can control gene expression. And it's also a connection to the journal article we looked at discussing biofilms. So now let's talk about quorum sensing. In short, it's a process of intercellular communication in bacteria through the interaction with small, diffusible chemical signals called autoinducers that can go on to impact gene regulation, and especially gene regulation within a biofilm community with high population density. So we have bacteria within a biofilm that are producing chemicals called autoinducers that then allow for communication between bacteria of the same species and different species. Quorum sensing allows bacteria to optimize their metabolic and behavioral activities, both within species as well as between species. It ensures proper growth and development of a biofilm, and as a group of bacteria get larger and larger within a biofilm, more of these chemical signals are sent out into the environment such that communication system is finally activated. We're controlling gene expression here. The release of chemicals, the autoinducers, result in an increase in synthesis of potential proteins involved in some kind of a cell product and some kind of a behavior. There are 4 steps to quorum sensing, and these are very straightforward. First, a bacteria is going to produce some small biochemical signaling molecule called the autoinducer. That's step 1. And then step 2, it's going to release the autoinducer into the nearby environment. Nearby bacteria, both the same species and the different species that might be part of a biofilm, recognize the autoinducers via receptors they may have on their surface, and there's usually some kind of a concentration threshold for this, a minimum concentration of autoinducers that must be met, and so this ends up being some kind of density dependent activity in terms of controlling gene expression. When threshold has been met, autoinducers induce some kind of regulation of gene expression. I'll be releasing a journal article on the overview of quorum sensing for you and for your reading pleasure. Not as a project per se, not as an assignment, but it's a short article that'll help you better understand the idea of quorum sensing. So this will be an exam type question. My intent both in including this here and in providing you with a short article to review is to give you a big picture of how intricate bacterial behaviors can be. We commonly think of bacteria as pesky little prokaryotes that cause bacterial disease, but as we move through the term, I think you're going to come to understand and appreciate just how beautifully complicated bacteria can be. And so with this is going to conclude the lecture on Chapter 7. If you have any questions, please don't hesitate to reach out. We can schedule a meeting by Zoom to talk about the questions you may have. Meanwhile, make it a great day.